Welcome. Welcome to Biology 1120. This is our lecture about the senses, in particular vision. And so my question to you is, do you require corrective lenses? Do you require corrective? And if you do, you can put your thumbs up. Put my thumb up. Okay. A few people do. Yeah. So quite a large proportion of the population does require corrective lenses. And they can be for various things. And we just saw a video on TED Ed about how glasses correct your vision. Some of us have it all. We have it all, but not in a good way. So I have myopia and astigmatism, presbyopia, and I've had cataract surgery. But you know what? The cataract surgery corrected the myopia, the astigmatism, and the presbyopia. So with cataract surgery, that is when the lens, because of aging, has stopped shifting to accommodate when something is close to you or farther away. And so nowadays, with the technology that we have in ophthalmology, you can correct for an astigmatism and for nearsightedness or farsightedness when you get your lens replaced in cataract surgery. So you're wondering why we have this funny looking little critter here at the beginning of our lesson on vision. It's very odd. Have you ever seen one before? This little drawing. You probably haven't because you know they're very small. It's a flatworm. And I put it up here because I like to introduce vision by talking about the evolution of the eye. Because the eye is fairly complex, but it evolved from simpler structures. And if you understand the simpler structures, sometimes it's easier to understand the eye. And so this flatworm is a little nondescript invertebrate, and all it has is an eye cup. An eye cup. So let me write a few notes about how the eye has evolved. So what do you need? What kind of receptors in particular do you need to interpret light? So the receptors, if you recall, transduce signals. Good, yeah, transduce signals. In this case, the signal of light to an electrical signal, an electrical chemical signal. That's transduction. And remember, transduction requires the membrane of the receptor to depolarize. And so how does the membrane of the eye change? You know, when you have a chemical, the chemical can bind to a protein in the receptor and the membrane will change and ions will flow in, changing its potential. But what about light? How does that work? What do you need for light to change the membrane potential in order that ions can flow across? Well, you know, it's interesting because what you need, you've heard of before, is called pigments. Pigments. Pigments are the king of vision, or the queen of vision, if you like, or the they drive it. They really do. They drive vision. And they're quite simple. Pigments are just molecules that absorb certain wavelengths of light. And the interesting thing about the pigments in your eyes retinal, it changes shape when light hits it. So these molecules change shape when light hits them, triggering the whole process, triggering transduction. So where the membrane potential changes and sets off a series of events causing a signal to the brain. Well, pigments are pretty simple, and all you need, like say you are a single-celled organism, like this one. Oh, I don't know how to get green on here. Anyhow, let's make this a little single cell. This is a single cell. This is one that exists today. There's many single-celled organisms that exist today in water, in our waters, in the oceans and lakes and rivers and anywhere where there's water. So this is a single cell, a protist, known as this one we'll say as a euglena. It has a flagellum, and something else very interesting. It has a little tiny eye spot. Of a patch of pigments. That doesn't change the membrane potential of the whole cell, but it changes the inside of the cell enough so that this organism knows that is the direction of light. So if light is coming to it in this direction, well, seeing as how this is a photosynthetic organism, do you think it will move away or toward the light for photosynthesis? Do you think it will move away from light or toward the light? Toward. Sure, it needs light for photosynthesis. So light hits the pigment changes its shape and alerts this little cell that it has to swim toward the light. Yeah, other animals like this one, it doesn't move toward the light. It's an invertebrate. For it, light means something can see me and conceivably eat me. So it moves away from light. But those pigments for this organism are concentrated. They're concentrated in cells that are very particular. So I'm just going to show you a layer of cells here to illustrate. Just so you can see how these cells may have changed over time in the evolution of the eye. So here's a layer of cells. Highly magnified, of course. And these cells have pigments. And these cells can alert an organism's nervous system about the direction of light. So say here's a fish. Oh, that's not a very good fish. That's a terrible fish. Okay, here is a much better fish. And this fish is swimming by, and light is reflecting off the fish. toward this animal. This is light coming off the fish, but it's not just hitting one cell. You know, the light from the fish is hitting all kinds of cells in all directions. There's nothing directing the light. This fish is swimming by, and these cells are picking up that something is swimming by. So the image that this animal is getting is probably just a shadow. It's probably just a shadow. So, you know, I'm largely exaggerating the cells in relation to the fish. That's just for illustrative purposes. So for our flatworm with the indented eyes, the eye cup. So here's the eye cup with the pigments. There's a membrane. So that's just a light membrane. And here's the fish again. But this time, there will be an angle that the light hits some of these cells. So because the cells are indented, it does give this animal, this might be the flatworm, a sense of how far away that object is. So it gives the animal a sense of shape, a little bit of a sense of shape and distance because of the angle of light that's hitting the cells at a different angle now, collecting some of that light. Well, here is another animal. So this example is the flatworm. There's a membrane. But this membrane has a hole in it. Those are pigments. And here's the fish again. This is the eye of the fish. Well, this little hole here makes all the difference to resolution or being able to determine the shape of this object, because light at the head end will go through that hole and only hit this cell here. I'm exaggerating a little bit, but light from the tail end will go through that hole and only hit this cell here. And light from the middle will hit the middle cell. So then this animal will get a sense of the shape. So shape of object is clearer. Although it will be reversed for the animal. And this could be, this is called a pinhole eye. And there's an animal that has a pinhole eye that you may or may not have heard of. It's called a nautilus. And it's a distant relative of the octopus, the pinhole eye. The flatworm has an eye cup. So the eye is getting more and more complex and causing images to be more and more resolved, better resolution. And so the last one I want to show you, we still have an eye cup. There's tissue at the back. So we're going to call this tissue retina. Here's the membrane with the hole. Here's the fish. But this time, this fluid or maybe a gel that's inside the membrane is solid here. It's more solid, connected by ligaments. It can change shape. So this object is clear but still somewhat fuzzy. But as soon, because, and the reason is that, you know, the light coming through here from this organism, some of it's not focusing on one spot in particular. It might be focusing in different areas. So it's clear but still fuzzy. But if you can get this light to focus exactly on the retina, that makes it very clear. But of course, still backwards. So clear still. And so this is called a lens and cornea eye. A lens and cornea eye. Very interesting progression from simple to complex, making objects less clear, but maybe being able to tell where they are by shadow, to resolving the detail on an object to tell exactly what it is and give it shape. So here's the nautilus. Its eye, it's got a little pinhole eye there. So these animals exist today. So it's interesting when you look at the evolution of structures of the human body, you can look at other animals and see where these structures were more simple and how they may have evolved over time. Another kind of eye that's evolved is the compound eye of insects. And they have little tiny lens and cornea eyes called facets. So each individual one is actually called an ommatidia. How do you spell that again? I forget. Ommatidia, I think. So this fly might have hundreds of ommatidia, but each one sees its area separately. Just like our two eyes see everything separately, our brain puts together what we're seeing. And this animal might see 100 things separately in its environment. So this is an image that somebody made on a computer to simulate what, say, a bee is seeing through a compound eye. So as you can see, not very clear. So not clear. But see how the ommatidia are distributed almost 360 degrees or 180 degrees. So this animal can sense motion so well. So compound eyes of a fly sense motion very well, which is one of the reasons we have peripheral vision. Yeah, we can see peripherally, and we can sense motion more readily with our peripheral vision. So have you ever tried to like hit a fly or swat a fly? You can't do it because the fly sees your hand coming like so fast. They can see a fan that's rotating. We might only see one image of the fan, but a fly can see every blade of the fan individually. Its motion vision is so good. Look at this dragonfly, 28,000 facets. Dragonflies are so good at catching other flying insects. They're amazing. If we were to get, though, the same resolution as a lens and cornea eye that we have with a compound eye, it would have to be this big. Yeah. So compound eyes, not good for resolution. Or another way of putting that is seeing detail. So by resolution, I mean, so if I were to draw two lines like that, You can resolve them as two lines from where you're sitting. If you moved way back, though, they would probably only look like one line. If I draw them very, very close together, like that, if you look closely, you would see them as two lines, but if you move back, you'll only see them as one line. Resolution is being able to resolve detail like that. Now, what's really interesting is that invertebrates like squid have evolved the same kind of eye that mammals have, the lens and cornea complex eye, but down a totally separate line of evolutionary history. So there's a squid have amazing vision as well. You know, even scallops have eyes. You don't think of that, but scallops have these little lens and cornea eyes. Here they are. This scallop has 60 little eyes that can determine what's in its surrounding, like say a sea cucumber, which eats them, and it will scatter away really quickly. Or you can eat scallops too. They're very delicious. So yeah, there's all kinds of eyes in nature. Spiders, interestingly, have both. They have simple lens and cornea eyes and compound eyes. Amazing. Okay, let's look at a human eye. Finally, right? Let's look at a human eye. Okay. So we will also dissect an eye. Let's look at some of the parts and see what their functions are. Superciliary ridge. So feel that. That's just above your eyebrow. There's a hard bone there. That's your superciliary ridge. And that's where your skull protrudes, giving you a brow ridge. The eyebrow here, of course, your eyebrow is a selection of hairs. What do you think is the function of the eyebrow? What's the function of your eyebrow, do you think? Sure, protection, yep. Filtering things from getting into your eye. Yeah, so from your hair and from your skin, there's dropping of cells and plus debris that might be falling from above. Exactly. And then there's your eyelashes. The palpebral fissure, pardon me, is the area between your lids. The lateral commissure is where your eyes come together, your eyelids come together. The conjunctiva, and we'll talk about all these separately, is a layer inside your eyelid. The pupil is the opening between your iris. So the iris surrounds the pupil. The sclera is the outer layer of the eye. It's quite thick. The tarsal plate is where you have glands that secrete oils onto your eyes so they don't dry up. There's your upper lid and part of your lacrimal system for tears. The medial commissure, so that's closer to your nose. Your lower eyelid. And your palpebral sulcus. So these are the sulci, the grooves that form under your eyes. Yeah. So vision is all about light. Vision is all about light. Sight is perception of light that is emitted or reflected from objects in your environment. The light that we can see, visible light, sorry, it's radiation, electromagnetic radiation, with wavelengths from 400 to 750, sorry, from, yeah, 400 to 750 nanometers. A billionth of a meter is a nanometer. Yeah. So light causes a photochemical reaction to occur that we talked about earlier. And produces a nerve signal. So the light has to be perceptible. We, for example, we don't see infrared. Infrared is heat. It's below 400, so you can't see it. All it does is heat your tissue, but it's not enough to cause a photochemical reaction. On the other hand, so less than 400 nanometers is too weak to cause a reaction, and greater than 750 nanometer wavelengths, you're looking at UV radiation, which is too strong. It damages tissue. Yeah, and that's why you should wear sunglasses when you're in the sun, or a hat, because UV radiation can damage the tissue of your eye. And indeed, it's thought that that's what causes cataract. So the reason that I got cataracts is probably because of playing volleyball in the sun for the last 10 years or so, and really sometimes not even wearing sunglasses, which was not great. I did a lot of the time, but sometimes they would just fall off. And yeah, so you should definitely wear a hat or wear sunglasses. So, yeah, before we go on, it's the other way around. Yeah, I'm sorry, I have to change this. If it's greater, I was going the wrong way. If it's greater than 750 nanometers, in other words, here's wavelengths of 750. But if it's they're large like that, it will just create heat. If it's less than 400, so say this is 400 nanometers. If it's less like that, that will cause tissue damage because they're so small. So smaller wavelengths cause tissue damage. Sorry, you'll have to rewrite your notes there. I'll just give you a second to do that. So the large wavelength, the amplitude is too weak. And so the premise of that is that if there's an object that's this large, well, these wavelengths will hit it, but these wavelengths will just go around it, essentially. All right. Let's look more closely at our eyebrows and eyelids. So eyebrows, they're protection from glare, perspiration, particles. So have you ever seen football players, they put those black lines under their eyes? So that's meant to stop glare. Eyelids, they block foreign objects also. They blink to moisten the eye. They meet at the corners, which are called commisures. They consist of a muscle. The muscle is called orbicularis oculi. And a tarsal plate with skin on the outside and conjunctiva inside. So muscle, conjunctiva. And a tarsal gland, or tarsal glands rather, secrete oil. That keeps the eye from drying out. It reduces the evaporation of tears. And the eyelashes keep debris out. Eyebrows and eyelids. Has anybody ever had conjunctivitis or know anybody that's had conjunctivitis? Let's look at that. Let's look at the conjunctiva. So here it is here. It's relatively transparent. Mucous membrane. The conjunctiva is a mucous membrane. It lines the eyelids. It secretes a thin mucus film over the eye. So that also helps to prevent from drying out. It's richly innervated. That means it has lots of blood vessels. So it heals quite quickly. Conjunctivitis is also known as pink eye. Yeah, thank you, Jen. Yeah, pink eye. It's caused by a bacteria or a virus. So it causes inflammation of the conjunctiva when it's treated by antibiotics, generally. It can be caused by dry eyes. It can be caused by allergies. So there's various causes of conjunctivitis. The lacrimal apparatus has to do with tears. Yeah. So what is the purpose of tears? Well, again, washing out debris from the eyes. So tears flowing across the eyeball helps to wash away foreign particles. Helps to wash away particles. It also helps with the diffusion of oxygen and carbon dioxide. That your cornea requires. And tears contain a bactericidal enzyme that kills bacteria. To protect your eyes further. That's called lysozyme. Lysozyme. So how are you at moving your eye around? Can you move them up, down, left, right? Can you move them in a circle? So you can do that because you have eye muscles. So the eye muscles are attached to the sclera, the outer tunic of the eye. And there are 6. So 6 muscles insert on the sclera. That's the external surface of the eyeball. 4 are rectus muscles, and they move the eye up and down and left and right. So there's the superior rectus muscle, the medial rectus, the lateral rectus, and the inferior rectus. So 4 of those. And the obliques. And they are the superior and inferior obliques. They're a little more complicated. They rotate the eye. Superior and inferior oblique. And these muscles are innervated by cranial nerves. They're innervated by cranial nerves, which are the oculomotor, trochlear, and abducens. So there's 3 cranial nerves that are devoted simply to the eye. So 6 muscles insert, that's that word there, insert on the sclera. Tunics. Another word for coat or covering or cloak. So there are three: the sclera, the choroid, and the retina. Those are the three layers of the eye. So the sclera and the cornea make up what is known as the tunica fibrosa. The sclera and cornea are continuous. There's the cornea at the front. The cornea bulges out. The eye is normally quite spherical in shape, but the cornea bulges out at the front. The sclera continues on to surround the optic nerve. So here's the optic nerve. And the optic nerve is continuous with the retina. So the retina, which is the inner layer here, is all neural tissue, and it's continuous with the optic nerve. The vascular layer, the tunica vasculosa, is the choroid. The choroid is continuous with the ciliary body there and the iris. So that's all one layer: the choroid, ciliary body, and iris. The internal layer, the innermost layer, is the retina and the optic nerve. So the ciliary body is a muscular ring and it's around the lens. The iris maintains control over the pupil. It has, so controls pupil size or diameter, and has chromatophores that give it color. Chromatophores. So if there's a lot of melanin, for example, melanin is the pigment of color. The eyes are brown, and the vast majority of eyes in the world are brown. If there's less melanin, the eyes are blue, and in between you get other combinations. And there are other factors contributing to eye color as well. So those, my friends, are the tunics of the eyeball. Let's see if I need to say anything else about that. Oh, the choroid. Yeah, the choroid is highly vascularized. It's vascular and pigmented. So when you see it, we'll show you a dissection. It's very dark and pigmented, so that when light enters the eye, sometimes it gets scattered off of other tissue inside the eye, and then it can be confusing. So the scattered light is absorbed by the pigments in the choroid, and also at the back of the retina. There's a layer of pigmented cells. The optical components are a series of transparent structures that bend or refract light rays to focus them on the retina specifically. So the optical components. When light is entering the eye, what is the first optical component? So here's the eye. So that's the cornea. Good, yeah, the cornea. And then just beyond the cornea is a type of liquid known as aqueous humor. What do you think the next structure is? Starts with an L. Is the next structure that light hits? The lens. So there's the lens. And then back here, there's also some gel type of liquid. It's more viscous than aqueous humor, and it's called vitreous humor. Not unlike haha kind of humor. It's just a substance. So the cornea is transparent, of course. Aqueous humor provides nutrients to the eye, the cornea. It supplies nutrients. It maintains the shape of the eye. So it has sugar, it has some vitamins, some proteins. But it's really, though, it's about 99%, at least 99.9% water. The lens. The lens is suspended by ligaments. So it can change shape because of those ligaments, which are also attached to muscle, of course. What else? Vitreous humor. Yeah, so that's kind of a jelly. It also helps to maintain the shape of the eye. And it presses on the retina. It presses on the retina, which is at the back of the eye. Good, okay. Moving along, aqueous humor. Here it is here. It's important to remember that it's in front of the lens, whereas the vitreous humor back here is behind the lens. Yeah, so it's produced by the ciliary body, produces some of it. Produces aqueous humor. Yeah. that's all I wanted to say about aqueous humor. Neural components. That includes the retina and the optic nerve. The retina forms as an outgrowth of the diencephalon. So the retina is the only part of the brain that you can actually see. So, if you've taken a picture of somebody and their eyes are red, they get red eye in the picture, you're seeing the retina essentially. And yeah, it started off as part of the brain, part of the diencephalon. So it's only attached at a couple of places. Other than that, it's just maintained there largely by the vitreous humor, although that's mostly when you're young. So it's only attached at the optic nerve and a structure known as the ora serrata. The ora serrata, which looks a little bit scalloped. So it's pressed against the rear of the eyeball by vitreous humor. So do you know anyone who's had a detached retina? A detached retina? Yeah. So, one thing about a detached retina is it's very important to realize it right away. So a detached retina, it's caused sometimes by blows to the head or by shrinking of the vitreous humor, which happens with age. So it's more likely to get a detached retina with age. So a friend of mine had it. The symptoms are flashing lights, particularly peripherally. Flashing lights that have no external cause to them. And sometimes, if it's a severe one, it can be like a curtain is crossing over the front of the eye. As it's detaching, and blurry areas of vision. So you may be looking out there at something, the trees, or you're looking at something on the wall, but part of it is blurry. Not the whole thing, part of it is blurry. But it's very important because to catch it very early because it can be reattached quite easily, but it has to be reattached in a short space of time. I'm not sure what that is. I thought it was 6 hours, but it's not, I don't think. I'm not sure exactly how much time you have, but not that much time. It can eventually lead to blindness. And that's due to a lack of blood. So the retina will no longer be getting a blood supply from the choroid, which is where the retina gets its blood supply. So these are the blood vessels that someone would see looking through an ophthalmoscope. This is the macula lutea, is this area here. It's about 3 millimeters. So it's not very large, but it contains the fovea centralis, which has most of your cones. So it's at the center of the macula. With packed receptor cells, particularly cones. So an examination of the eye will show the condition of the eye based on the blood vessels and the macula, which you can see here. Let's see if you can do this test. So the blind spot, also known as the optic disc, is where the optic nerve exits the eyeball. And because that's happening in a particular spot, there are no receptors there. So at the back of your retina, where light is hitting your retina, there is an area where no light will hit. There are no receptors. That's called your blind spot. So this is a test for the blind spot. So you're supposed to look at the X from a certain position, and then the red dot will disappear. So I think you have to stand back a fair bit. So if you close one eye, in this case, you'd have to close your right eye and look at the X. It depends where you are. You have to be in a very particular spot for that to work. But you can test it yourself online. What happens is your brain just fills in the red spot with the green line because it just figures that must be what's there. So that's the blind spot. So I'd like to just look at refraction quickly. What is refraction? So here is an object. At 90 degrees, that light's just going to go straight through. No bending. But if light strikes at an angle, then this object, whatever it is, will bend the light a certain amount and it will be refracted. So you could try this in the bathtub. If you're in the bathtub and you put a ruler into the bathtub, you'd expect to see it at a certain spot, but the water bends it. So the bending of light occurs when light passes through a substance with a different refractive index at any angle other than 90 degrees. So arbitrarily, the refractive index of air is set to 1. So n equals 1. It's just an arbitrary number. It could be anything. So what you do is you compare the refractivity of other objects. You compare that relative to air, or 1. So the cornea is 1.38, and the lens is 1.4. So that's just an indication of how much the light is bent. And that's very important for being able to resolve images or getting fine detail, because that will only happen if light is focused on the retina, because that's where all your receptors are. They're all on the retina. So it's no good if the light is behind or in front of the retina. That's not helpful. So various things happen to the eye. Emmetropia is when you're looking at something far away in the distance. So the object is far away. And because it's far away, the light enters the eye perpendicularly, just like that. But if something's close up, then your eyes will have to focus on it. So the first thing that happens with a near response, is that the eyes turn inward to converge on that object. a close object. And the other thing that happens is with the pupil. So with emmetropia, when something's far away, you're gaining a lot of light from the distance, and your pupil is relatively dilated. If something is close up, so this is something far away, this is something close up, the pupil is relatively constricted. So that stops blurry edges from occurring. It's a spherical aberration. When light enters the eye, if the pupils are very large, it may appear blurry. So it does not, doesn't admit peripheral, in other words, coming from the side, light rays. And it reduces blurriness, or aberration it's called, blurry edges. Sometimes you can see that when you're looking through a telescope. And what about the lens? If something's far away, the lens is relatively thin. If something's close up, the lens is relatively thick. So why should that be? Well, a thick lens bends light more. To focus on the retina. So sort of the natural condition when the lens is thin is that the light will be focused on the retina. But if something's close up, the angle of light is more acute. And so it needs to be bent in order to land on the retina. So here you can see the light is parallel, but here it is not parallel. So it has to be bent more. And that is the function not only of the cornea, but also the lens, which can be adjusted. The cornea can't be adjusted, but the lens can be adjusted. So it relaxes into a convex shape. More convex. And that occurs when the lens is thin. The muscles, the ciliary muscles, are relaxed, but the ligaments are tight or taut. So they pull on the lens, keeping it flat. So that's when something is far away. But with near vision, with accommodation, the muscles contract, the ligament is relaxed, and that causes the lens, instead of being taut and tight like that, causes it to relax into a rounder shape. And so as we saw with the video, if our lens isn't doing the job, well, it's actually not our lens that's not doing the job. With myopia and hyperopia, is because of the shape of the eyeball. So in the case of myopia, so this is myopia here. So uncorrected, look at where the focal point is there. And that's in front of the retina. So the type of lens that you have to have is a concave lens, your corrective lens. That's thicker on the outside and thinner on the inside. And that causes light to bend a little bit more so that it will focus directly on the retina, the concave lens. It causes the light to bend outward. And the opposite is true of hyperopia. So in this case, the eyeball is too short and the focal point is back here. So that has to be bent more. In order to bend light more, you need a more convex shape. So this is a convex lens that will bend light more. So I'd like to stop there. Thank you for watching.